Sawadika. Good afternoon, ladies and gentlemen. First of all, I would like to thank for organizations of ICP today gave me the good time to present on this conference. And today I will present a part of my study as a Ph.D. program. Please wait me a minute. And could you hear me and see the screen? Do you hear me?
Yes, that's right.
Okay, thank you. I am Nguyen Thị Hải. Now I finished my program at Kaen University, and I worked on departments of parasitology in Thailand University in Vietnam. And today I will present my study, a part of study with title Alteration of Gut Microbiota in Strongyloides, stercoralis-infected chronic kidney disease patient in a community. This research under chronic kidney disease, not in Thailand, and Faculty of Medicine. About the content of this study. The first is the background and rationale, the objective, experimental design, result, discussion, and conclusion. And so about I would like to show you I have one question where I study what is the relationship among stercoralis and kidney disease and gut microbiota. And as we know, in North Thailand is an area endemic areas of Strongyloides, stercoralis, and here is a with high ratio of chronic kidney disease. And also many research now shows that gut microbiota had growth on the kidney disease as in advanced progression. So I would like to answer this question. What is the relationship among this? And I would like to find the answer. First of all, I would like to show about Strongyloides stercoralis. Strongyloides stercoralis is as a phylum nematode, and then Strongyloides species stercoralis, and Strongyloides contain at least 15 species, and stercoralis is a major cultivar Strongyloides in human, and another race human infecting species that is Fullenbori and Fullenbori subcaliis. And about in North Thailand, a researcher, a professor Wanchai Malung, you can see in the picture with the stercoralis in the North Thailand with red color, more than 20.2%. And I studied on Khon. You can see it with the yellow color, more than 40%. And stercoralis has a complicated life cycle, as you see on the screen, with the three kinds of life cycle. The first is the auto-infection. They can develop by the cell in the host. And the second, the free living cycle, they can develop in the environment without host. And the third is the natural infection with filiform larvae. They develop in the soil and infected in human when they enter the circulation of human and break through alveolar and swallow on intestinal and complete the development in intestinal. And with the acute infection with diarrhea, constipation, abdomination pain, and gastrointestinal cutaneous manifestation, and special more severity is with stercoralis infection can cause hyperinfection syndrome or disseminated and cause another disease severity. And about another disease is a kidney disease. This is one kidney cross section. The kidney disease can occur when kidney damage or injury of the structure and functional abnormality by stimulus factor, and kidney disease can classify two kinds, with the damage or injury less than 3 months. They call acute kidney disease, and another more than 3 months, they can chronic kidney disease. And with chronic kidney disease has many cause and risk factors. For example, inflammation, high fat, high fructose diet, diabetes, high blood pressure, heart problem, obesity, farm history, tobacco use, or error, or use alcohol. And about in the world, one research showed 2050, the ratio chronic kidney disease in the world, you can see with the red color, it's more than 2,000 per million population. And I note that in Thailand, chronic kidney disease is a yellow color, is a circle with 5 to 1,000 per million population. And the chronic kidney disease were classified to five states based on the glomerulonephritis rate. This is a glomerular rate and ratified from Z1 to Z5, with Z5 is a kidney failure. As about the, when I study, I think it's about what is the relationship with Strongyloides, stercoralis, and kidney disease. And I review many papers. They show the stercoralis infection and kidney disease with oral corticosteroids, and I found that 18 cases of Strongyloides stercoralis associated kidney disease, in which 11 cases survivor and 7 cases die. So I thinking about this link together. And another about the gut microbiota, as I mentioned about before. The gut microbiota has 3 kinds beneficial bacteria. They can maintain a health, and another opportunistic pathogenic bacteria, they harmless in their habitat, and harmful bacteria, they can impairment of health. And the gut microbiota linked with kidney disease, as shown in this chart. You can see in this map, with the dysbiotic gut microbiome creates the endotoxin, for example, pure-creation sulfate, endotoxin sulfate, or immune dysregulation can cause the infection, and all of this can cause CKD or another disease. And so I link with another Strongyloides or helminth. They link with gut microbiome, and many research show that helminth can alter the gut microbiota. So I would like to find the answer whether stercoralis infection alter the gut microbiota and relates to CKD. To answer this question, I found one way, that is metagenomics. Metagenomics were used to study the interaction between parasite and gut microbiota. Metagenomics collects the genome of microorganisms from an environment sample to provide information on the microbial diversity, ecology of a specific environment. And that I would like to show you the workflows of metagenomics. And DNA will collect isolation and DNA amplification and DNA sequencing. After that, bioinformatics and data presentation. And I would note that DNA sequencing was used by high throughput sequencing technology Illumina platform. And I would like to explain more about this. With the first step, the fishes will collect and extract the DNA, and the DNA will amplify based on the genes of 16S RNA, and the sequencing of this, and group to one OTU one OTU based on the 97% similar sequence into one OTU, and after that, data analysis. And about the 16S RNA then sequencing, I would like to explain more about OTU. OTU is one operational taxonomic unit. That is one unit. It's classified 97% similarity of gene in OTU based on 60S RNA. You can see in the screen, this is a 60S RNA from V1 to V9 region, and based on this region, we create the library. And almost this is V1, V4, V3, and V3, V4, V4, V5, and they can also sequence with V4 or another. However, when I search and I found V4, V3, and V4, it shows the high diversity. So this study, we use V3 and V4. And about the DNA sequencing using the Illumina platform, I will show you all the steps. With the first step, a library preparation. Next generation sequencing library is prepared by fragmentation a genome DNA sample and ligating specialized adapter to both fragment end. And after that, they cluster application on the flow cell. Library is loaded into the other flow cell, and the fragments are hybridized to the flow cell and in the surface, and the correct amplify into a clone cluster. Here is a clone cluster. And after that, they will sequence based on all this cluster, and they saw which the size signature connect with computer, and they saw all the cluster and all one cluster with its read corresponding. And after that, alignment and data analysis, all the reads can compare with the reference genome, and they show the which corresponding the reference genome. That is about genomics with Illumina, and I would like to show you the objective in this study. This study was to investigate the effects of Strongyloides infection on gut microbiota in CKD subject. About the experiment design, from 230 CKD, we collect the feces, blood, and separate. The Strongyloides stercoralis were identified by ethnicity or agar plate culture, and we separate with stercoralis positive and stercoralis negative. And we match pair about sex, age, and another factor. We choose the 20 samples suitable for X, and the total is 40 samples. And all the sample negative, we check with conventional PCR with negative Strongyloides stercoralis. And after that, individual species sample will do with next generation and sequencing create the metagenomic profile. And the result come out. About the result, we saw in our study, based on previous study. Previous study, we saw that the average of CKD state is higher than only Strongyloid in CKD. CKD and Strongyloid with CKD state higher. You can see in the red color with orange more than one, however, not significant. And you can see in the table with negative with Strongyloid only have 3 states, CKD 1, 2, and 3. Oh, sorry. However, in Strongyloides with CKD, we saw with stage 2 and stage 3 is higher, and so the stage 4 have 2 cases. That is a characteristic pattern. All the factors we must pair with the 2 groups, Strongyloides negative with CKD and Strongyloides positive with CKD, and the result so not has a difference about the male sex, age, and another factor. This is the similar factor. And another result show the different alpha and beta diversity between Strongyloides positive and Strongyloides negative. And you can see in the picture A, Sanong. Sanong is one kind criteria for the diversity, the alpha diversity. They show the alpha diversity, and you can see Strongyloides negative is higher Strongyloides positive diversity with CKD. And also about the beta diversity. Beta diversity in the figure B, you can see which Strongyloides negative is higher more than Strongyloides positive and CKD. So we can conclude that about the Strongyloides positive and CKD, they show alpha diversity and beta diversity lower than Strongyloides negative with CKD. And another principal coordinates analysis, PCOA. And PCOA is this figure, so with separate X point, you can see with red point is the Strongyloides, the sample of Strongyloides positive, and green point is a Strongyloides negative. And this picture shows that separate point, it means the different communicates of bacteria in each sample. And another linear discriminant analysis. This figure shows the detect biomarker with the statistics different among group. And you can see which Strongyloides, all Strongyloides positive with red color. You can see Escherichia sinella, Escherichia coli, and some another family or order. And another Strongyloides negative, you can see with green color, only one order Bradymonadate. That is a biomarker. And this figure shows good gut microbiota composition between Strongyloides positive and Strongyloides negative, with in the level phylum, class, order, family, and genus. And you can see in the phylum it's not have too much the difference. However, another class, order, family, or genus, you can see the different color in each column. And for special, you can see in the genus, figure E, genus for Escherichia sinella. Escherichia sinella is one kind of bad bacteria. And you can see which Strongyloides negative on CKD is very low. However, in Strongyloides positive in CKD is very high, significant difference. And another, you can see in this figure shows the significant difference about two groups, Strongyloides positive and Strongyloides negative, which Strongyloides positive is the orange color and negative is a blue color. And I will note that Escherichia sinella is very high in Strongyloides positive with CKD and lower in Strongyloides negative with CKD. And another Rotia. Rotia is one kind of bad bacteria, the same Escherichia sinella is a bad bacteria. It's the increase in CKD with Strongyloides. But another anaerotype is a good bacteria. However, this lower, they reduce in Strongyloides and CKD. And I would like to show you in our research, Our research so have two bacteria, two xenotes of bacteria are opposite chain. That is anaerobic good bacteria, and pathogenic bacteria is Alteration. You can see they different opposite chain about the egg, sex, and CKD state. This is A and B, figure A and figure B, so with the age. That is opposite chain. I classify to lower 65, 8 years old or more than 65. And you can see the significant difference of this chain with anaerobic is very low and Alteration is very high with Strongyloides and CKD based on its lower or higher 65 years old. And another with shake. You can see the number abundance of anaerobic is the same with age. I classify male and female, and the results so they have different between Strongyloid negative and positive. You can see here with Strongyloid positive, Strongyloides is very high in Strongyloid positive and low in Strongyloid negative. And special with female, Strongyloid positive and CKD, very high abundance of Alteration. And about they different about the CKD state. And you can see in this figure about anaerobic good bacteria is very low in each state. And different that Alteration is high. Oh, sorry. It's high in CKD and Strongyloid positive. So we know that anaerobic and Alteration are opposite chain in CKD and Strongyloid. About the discussion, CKD stercoralis infection on CKD, they can measure opposite chain with good bacteria reduce and increase the bad bacteria. Good bacteria, for example, anaerobic, Anchemeracia, Coprococcus, and another. And bad bacteria, they show some bad bacteria increase, for example, Alteration, Rotia, Streptococcus, and Haemophilus. And many studies show that important microbe in maintaining intestinal metabolic balance. All of these good bacteria have maintained the health on the human, but this bacteria deposition in many disease. And another, with increase the bad bacteria, they can cause the metabolized toxin increase and cause the gut disease and disease. And another, with bacteria related to CKD, we found good bacteria like anaerobic and Alteration and bad bacteria and Rotia, they can relate with CKD. In animal model, xenot anaerobic has a negative correlate with kidney parameter, and another E. coli and enterobacteria was markedly increased in paired kidney, and Alteration were positive correlate with metabolite toxin like TMAO or IS, and Rotia also with severities of kidney damage. About the conclusion, the first is candidate biomarker. With candidate biomarker, chronic kidney disease with stercoralis, my study shows the result. Candidate marker with stercoralis and CKD, that is E. coli, species E. coli, and xenot Alteration, and another xenot is a dilister. And about the chronic kidney disease, that is order Bradymonales. And in our research, so Strongyloides enter the gut microbiota and reduce the good bacteria, increase the bad bacteria, and reduce the bad bacteria and cause diseases in the gut in CKD. And another, they decrease the bacteria producing short-chain fatty acid and increase the bacteria producing ureate toxin, TMAO or ES, and they can cause the advanced CKD progression. About the strength and weakness, the strength of this study, the characteristic of two group control and stercoralis infection with pair matching sex, age, and factor. However, they have many weaknesses. For example, the sample size was small, or just so the TMAO or IS producing bacteria, however, did not measure the insulin in sample. And lastly, I would like to thanks to all of you FP lab member. Also, of you helped me a lot in my Ph.D. program when I study at Khon University. Thank you for all the participants. Thank you very much.
Thank you very much, Dr. Hai. Now this is the question and answer session. So we have question on the floor in the chat box. Please explain more about how to select the pair volunteer to compare the effect of Strongyloides positive and negative on CKD patient.
Thank you, Joe, for your question. I would like to answer this question. When I choose the participant, I have to matching with the participant Strongyloides positive and negative based on the sex, age, and another factor. And to compare, I have many criteria and blood index. And when I matching the participant, I have to choose the same. For example, age, I would like I choose the same age, or special sex is the same. And another factor, for example, the disease, they have which Strongyloides stercoralis positive have the obesity, and Strongyloides negative also the same index, and another criteria is the same.
Thank you. So are there any questions from the floor? You can raise your hand. Oh, there's another question in the chat box, Dr. Hai. So do you think that Strongyloides infection alter microbiota and subsequently cause severe CKD? Is that correct?
To conclude that, I need many research, but this is one kind. Through this research, I would like to show you another way to research about interaction between parasite and disease. And not in Thailand, have enemies with Strongyloides and high chronic kidney disease. So when I link with this and investigate the gut microbiota, and I saw many different about this, significant difference, and I saw good bacteria in Strongyloides, which good bacteria reduce in Strongyloides and CKD, and pathogenic bacteria very high in stercoralis with CKD. So I think that based on alpha diversity and beta diversity, I saw they have changed. So I conclude that Strongyloides in CKD maybe can alter the gut microbiota.
Also, another question in the chat box. Nice presentation. I have a simple question. Why there are only a significant levels of genus Escherichia shigella in CKD patients over 65 years old infected with stercoralis? Is there any report in the young age group?
Yes, thank you. Because based on my data, of my data is have when I classify, I have to choose the O, and when I classify and I separate many another but not have significant, and when I choose with 65 and they have significant difference between them. So, and I don't find any another journal and not find, yes. So if you have another journal, so that maybe could you send me, please?
Thank you very much. Are there any questions? There is no question so far. So if there is no question, we will move to the next session. Thank you very much, Dr. Hai. We hope to see you again. Happy to see you again.